Okay, so thank you very much. It was very exciting until now, and so let's see another model organism now, flies. And I'll be talking about, well, actually what I hope I can do is maybe take out the question mark. I don't think we need a question mark here, at least at the end of the day. And so we work with epigenetic regulation of development and the function of these proteins that I will introduce briefly to you today. So these proteins have been first discovered as regulators of development of the anteroposterior body plan. In flies, they are conserved, so you find them also in plants, mammals, and most of the species of the higher eukaryotes at least. And so what they do is they maintain the regulation of important patterning gene during development. So the way in which we set up our anteroposterior body plan, this is just a fly embryo that will turn up into the adult here, is to express specific genes that are called Hox genes in specific segments of the body. And so these genes have to be expressed in the right place, but also repressed in all the other places of the body. And so this is first made by specific transcription factors that are going to bind the promoters of these genes and switch them off. But then during development, what actually happens is that these Polycomb proteins take over, so they replace the transcription factors, and they recognize the silent chromatin and keep it silent throughout development. There's also another class of proteins called Trithorax group that does the opposite. It recognizes the on state in the right place of the body and helps maintain it on again throughout development. Now, years ago, we were actually, still when I was a postdoc, we realized that these proteins are not only capable of maintaining somatic memory, but also can sometimes perpetuate this memory through generations into the progeny. So we used again a transgenic trick to show that you can get this to work, and we were able to show that you can have at least a partial transmission for a few generations of such an epigenetic effect. I remember actually that Vincent, when I came to France, then I got this CRS position, and Vincent was telling me, why don't you keep working on this? It's really exciting, but you should really nail it down. And actually we tried, but molecularly it was very tough because it was a partial phenomenon. It got lost in most of the progeny, and we didn't have the molecular tools that we needed. So I today will show you that now we have the tools. So how does one of these transgenic lines look like that we established in the lab? And many other people have done similar things across the world. So what you can do is you take a binding site for these proteins, and instead of looking at it in the endogenous gene, you now hook up some reporter genes. So this is the binding site, never mind the name. Polycomb will bind there when you put it anywhere you want in the genome. And so if you have a reporter gene that is going to make red eye pigment, this gene will be shut down, at least partially, by Polycomb protein. So now what you see is that your eyes, the eyes of the fly, are not completely red. Instead, they are mostly white. And then they have these spots showing that the memory of this off state is not really perfect, in fact. So when you make transgenes, they are not as good as a bona fide endogenous gene. And actually you have a variation in the population so that you can find most of the flies in this range, like few cells that escape or very few of them that escape. But then you also have a few extremes, some cases in which you almost have a red eye and others in which almost everything gets silent. And we were very interested in studying that. We also realized something that was very surprising a few years ago, which was, so you have this transgene, you also have the same sequence that binds Polycomb here is also present in another chromosome, in this case, close to its target gene, the endogenous gene I was talking to you before. But we realized that actually these two talk together. And when I say they talk together, they communicate, what I mean is that if we make DNA fluorescent probes and that can hybridize to these two loci, and this is now an individual nucleus, what we can find is that the transgene finds the endogenous locus and they are frequently colocalized in the nucleus. And that was something very mysterious that we wanted to understand. So what we have been doing in order to study whether we could induce some kinds of transgenerational inheritance phenomena was the following experiment. So we have our transgenic line, it has the transgene, it has the endogenous sequence, but we also established next to it a syngenic line that has the transgene. The only difference is it lacks the endogenous source of sequence. And actually, in a line like this, what happens is that the transgene gets a little bit de-repressed now, because you're missing this contact that somehow helps silencing. So when you don't have it, you have a little bit of a loss of silence. So you will have a bit more of these kind of flies here. Now, what we do is we cross them together. We get an F1 that has the transgene again, homozygous. And now the endogenous sequence is missing only one of the two chromosomes. So this is a sort of limbo. It's a sort of intermediate situation. These flies can be back-crossed to the parental genotype. And we can obtain an F2, which is in principle identical, or as close as possible, genetically, to the grandparents. And as you see here, we actually also get this kind of spread in the phenotypes. Maybe the only thing we see is, again, a little bit more a tendency towards a little bit de-repressed kind of state. But, nothing really exceptional. When you look at the flies, say, well, maybe, I don't know if something really happened. But what we can do now, and with what we did, is to take the flies that are most silenced and cross them together for several generations. So every generation we discard, if we find these kind of flies, we take them out. And we keep crossing the very repressed individuals. And what do we get? What we get is a lot of flies that look like these, very strongly silenced flies. What happens if we now take the other extreme of the population, and again, we play the same game, but now we discard the repressed guys? Well, now we get very active ones. So how does it look? I'm showing just one eye. So how does it look when we look at the population? This is the starting population. Most flies like these, a few like these and these, almost nobody like here, and actually nobody completely active. But if we do this kind of selection, now we go into almost 100%, like 99 point something percent fully active, and 85 or something percent fully repressed. So actually, I can show you a bit of the quantitative version of the same experiment. So basically, we do this selection and we follow each generation. We're going to score the number of flies that have this phenotype or this phenotype. And as you see, initially, we don't see these very extreme cases. But then, with few generations, you see with the upward selection towards the active, we see the phenotype appearing, and the downward selection, it doesn't happen. Now, this is actually probably the most important part of the experiment, which is when we do the same selection scheme, but starting with the naive transgenic line. That is a line that has never seen this kind of crossing scheme when it was first destabilized in the contacts and then the sequence was put back. And we can try to do an upward or a downward selection. Nothing happens, absolutely nothing. So we never see the extreme cases. Okay, so that's what makes us think that what we see is really epigenetic. It's not due to some kind of sequence variability in the starting population. So now that we have established this, what is the genetics, if you want, of this epigenetics? So what are the laws that determine the transmission of this trait? And so basically, to put it short, the rules are defined by paramutation. They are following exactly what people have defined in plants as paramutation. And let me show what it is here. So what we have here is our Epialleles, our transgenic line that has gone all these maneuvers, and now it has an active Epialleles, so their eyes are red. This here is actually a kind of naive line, so you see that the eyes are kind of intermediate. But we made a trick. We put a marker here. You see the body color is a little bit darker than this one. And actually this is because we put a marker, a genetic marker, which is close to the transgene and makes this body visible to us. So if we cross them, these two, what we're going to get is we expose the epiallele to the presence of a naive allele here. And so we are in a kind of, if you want, heterozygous state. Then we cross these flies together again. And now they segregate in a Mendelian fashion. And so what we see is that when we take the flies that were like these, you know, that basically have reacquired the 2 copies of the original epiallele, they keep an active, actually this is not the same fly, yeah? They keep an active eye color. Okay, so they are still red-eyed. That you could expect. But what was very interesting to look is what happens to the flies that have now 2 copies of this one here. They should be naive, right? Because that was a naive epiallele. But now look at this, the eye is red. So that means that this naive allele has learned to be an epiallele. Okay, so now we can play the reverse game. We take these flies and we keep crossing them further. And we ask, is this one, this epiallele now, able to teach a naive allele again? So it has learned to be an epiallele. Can it become a teacher? So, okay, so it has learned to be an epiallele. But now, can it instruct another naive allele? And so here, these flies have a kind of this black marker. We cross them to the flies that have no black marker in the body and have, you know, this kind of variegated eyes. We do the same scheme, and what we get is, again, red-eyed flies. So this epiallele can teach, okay? So, and this is the exact definition of paramutation as it was defined in flies. But bear in mind, we don't have DNA methylation at the very, very low levels of DNA methylation, trace amounts in flies. So now we want to test here an essential prerequisite of this transgenerational inheritance, which is can we have reversion of this phenotype? And if we have it, we would expect, if it is epigenetic, it should revert at a much higher frequency compared to DNA mutation, because that's what has been seen until now in epigenetics. And actually, we can erase this epiallele in 2 ways. One is, I told you we start the whole process by playing with these contacts over long distances in the nucleus. Okay, so we took out the endogenous source of sequence and then put it back. So now, we have an epiallele that has also the endogenous source of the sequence in the body. So what happens if we take it out again? So here we have this white epiallele in this case. I mean, the results are always valid for both. I just show you one. We take this white epiallele, we cross it to a naive line, but a line that doesn't have the endogenous source of sequence. Okay, so we get first an heterozygous state, so the endogenous source is only heterozygous. Then we keep crossing, and we will get flies that re-establish the initial phenotype. So they have the endogenous now, because they were heterozygous here. So, and they keep the epiallele, no problem. They stay heterozygous, they also keep it. The interesting case is those that have completely lost both copies of the endogenous. What happens? They reset completely. So these flies, because they don't have the endogenous source anymore, they forgot, this epiallele forgot that it was in epiallele. It went back to the normal sort of intermediate state. So this is one way in which you can reset our epialleles. Second way is, I told you these epigenetic lines that we made, they bear transgenes that are homozygous. What happens if we cross this homozygous epiallele to a line that doesn't have any transgene, so they are white-eyed flies now? So we get first an heterozygous, which has a sort of intermediate eye color. Cross it together, we get 2 copies again. So do we get red eyes or not? So first of all, the results that are not interesting, we can reestablish the white flies that have no epiallele. Again, the heterozygous, no problem. This is what it should be. But how about the homozygous? Completely reset. They lost the state. So every time you lose some form of long-range interaction between the epialleles, you lose the state. What happens molecularly? So first of all, what we see reflects expression differences, RNA expression, okay, that's what you would expect. So if we look at the epilines in terms of expression of this white marker, the intermediate state is intermediate, the red is more expressed, that you would expect it. We also have a second report actually in these transgenes, which is just beta-galactosidase, like Z, and it follows the same rules. These are controls, they don't change, and a non-expressed Polycomb target gene doesn't get expressed, and an expressed Polycomb target gene stays expressed. What is important to note is that we see these things in adults, in which we take the heads and we see the eye color, but we can also, of course, look in any other tissue, and we took in particularly early embryos. And in early embryos, these genes don't have a promoter that is expected to express. Actually, there's no embryonic specific signal that dictates expression, and in fact, we don't see expression. We see background levels, at least, of RNA, so no detectable expression. And this is a very important tool for us because now it gives us the possibility to look whether we see something in the chromatin of these epialleles that are the same. They are not expressed in any case. Later they will be expressed, but they are not expressed now. So if we look at the Polycomb mark, it is this histone K27 trimethylation. Well, first of all, we look in the heads of the adults. We would expect to see something, right? Oh, no, sorry. Actually, I'm running ahead of myself. In fact, first, let's look at these embryos that have no differences in expression. But as you see, there is more Polycomb marking in the repressed epiallele than in the active epiallele. There is some Polycomb actually, because a gene that is not bound by Polycomb will have no marking at all. So we still maintain some levels of the mark, actually significant levels, but there are differences. And these differences will actually be maintained throughout development and even amplified because they become bigger when you take a tissue that is now expressing the differences in form of the reported genes. So to summarize this, we can say that when we have this kind of long-range interactions between an endogenous locus and a transgene, and we perturb these contacts, actually we can now offer a possibility to select towards a sort of epiallele that has a very strong repression or actually a very strong de-repression over generations. And this is leading to divergent phenotypes that are of fully epigenetic nature, and inheritance is stable. So we keep these lines. We don't need to apply any form of selection now. We keep them for almost 50 generations now in the lab. But this is all done in the lab. Can it be maintained in environmental, more like natural conditions? So we didn't really put these transgenic flies in the wild, as you might imagine, but we can mimic the wild condition in this ecotron, which is something that in infrastructure we have of the CNRS in Montpellier. So what we have is we put flies in very large cages with natural plants on natural food, and then we can change the climate, humidity, temperature, and we mimicked something that you would have between March and August. And then we ask whether these epialleles can be maintained or not. So this is just to show that you have a lot of stuff going on. You have lots of other species, soil flies, centipedes. Actually, some of them are predators of the eggs, and they killed one of the experiments. So we do that. We have a high variation of the conditions in the cages where the flies are, so they can actually select and try to find the best conditions for themselves. And when we do that, you see this is a triplicate experiment. Actually, this is a duplicate because it died because of the centipedes. This is the starting population, which maintains as it is more or less in the lab. And you see that the two epialleles can actually be maintained very nicely. Although you have a little bit of a loss, actually, of the active one compared to the lab situation. So the environment can actually impinge, at least partially, onto the epialleles, but doesn't lead to erasure. So, and to finish, can we see something like this, but without transgenes? Because, transgenes are always something a bit funny. You would like to see that in natural genes. And actually, we knew of a case, when we look at tox genes, that could perhaps be amenable to such a paradigm, because we knew that long-range contacts are not only involving transgenes, they can also be found in natural Hox genes that are in different loci in the genome. These two Hox loci, Antanapedia and Abdominal B, are spaced by 10 megabases in the genome in the same chromosome of the fly. And we know that they can contact each other, again, by DNA probes. We see this colocalization in the cell nucleus when they are repressed. So we could actually unpair these two by making a specific mutant in one of the two Hox genes. And the nice thing was that now when we look at the expression of the other gene, we could see phenotypes that are basically this emergence of legs instead of antennae. This would be a normal fly that should make antennae, but if you overexpress this gene, you start to make legs. And you make them not just when you mutate the gene, when you induce the expression of this gene by specific mutations, but also when you mutate something 10 megabases away that should regulate another Hox gene. And this is because you lost these contacts that depend on Polycomb and that help silence these genes. Okay, but now, because we've made these lines, what happens if we switch back? So we can make a cross where we first establish our lines that have these phenotypes. We can actually classify them in weak, moderate, and strong. Then we take out again the distant mutation that is 10 megabases away. And so now we reestablish genetically a line that should be identical to the starting population, okay? So actually, this is to show you, in terms of phenotypes, what is this starting population, okay? Which has moderate phenotypes, or sometimes a little bit stronger phenotypes, or actually weak or moderate phenotypes. These purple bars are the strongest case, which is when we have the Antennapedia allele, as it is, plus the 10 megabase mutation, okay, the mutation 10 megabases away, which now makes a lot of very strong phenotypes. And all these other blue bars are the situations in which you take out the mutation 10 megabases away. So you should be in the same situation like the black, but you're not. You have much stronger phenotypes, and they are maintained for 5 generations, as you see. So this basically shows that we can have stable epialleles that are not dependent on transgenes. So this is just the conclusions that repeat what I said, but the idea is that normal phenotypes are probably canalized into very stable states. Actually, we haven't been able until now to challenge them by just taking flies and change the environmental conditions. However, when you perturb, and in this case it is nuclear architecture of these flies, by changing the contacts at long range that happens between chromatin sites, you can trigger stable transgenerational inheritance of either silent or active states. They can be reverted to a naive state by, again, changing the 3D contact patterns. And this can be stable in relatively stable, although modulatable, in a natural environment. And inheritance involves transmission of Polycomb marking. So we believe that this is at least at the source of what we see in this particular situation, but maybe there are other regulatory forms in which you can obtain these similar phenomena. So this is pretty much the star of the project. Of course, it was done in collaboration, and many other people worked in that. In particular, we collaborated with Renato Pauer at the ETH of Zurich, and in Montpellier with Simon Fellou, Franck Sereb, and Jacques Roy, and the Ecotron. So thank you very much.
Thank you. Questions? Yes, Eva.
If you insert additional copies of the FAB7, do you reinforce the transmission, or is it interfering?
Yeah, this is something that intrigued us a lot, and we have to discriminate. When we insert additional copies in another transgene, we increase the power of silencing, so they get more and more silenced, actually. However, when we study specifically the transmission, what we can do is we replace the endogenous source with a transgene. We play the same game, but it doesn't work. So starting this endogenous locus is a locus where you have about 20 binding sites for Polycomb. So what I showed you is deleting just one of them, but it is a complex regulatory context that we have, and which we don't know why, but it is important for transmission.
Sorry. When you were showing the differences with the early embryo, with the modification, at that time, do you already see the change in the 3D organization?
Yes, but we are not yet at the super early stage, and we are looking into that, but I don't have the answer yet. I would actually doubt that. I think that there's a window of time in the embryo, which is the very, very early embryogenesis, in which in flies you have one cell division every, well, actually nuclear division because it's syncytial stages, every less than 10 minutes. And in this situation, we haven't done fish, we have done IC. And we see, very little signs of any specific architecture. But this doesn't mean that there is no inheritance. What could happen is, because actually what we see is, as soon as we see a little bit of structure, which is still in the syncytial stage, the first thing we see is a contact between the Hox loci, which is incredible. I was really shocked when I saw it. So we are reproducing this. But what we believe is that there might be a contact in the oocyte at least, and this would be perhaps transiently not visible while you are having these very fast replications, but then, as soon as the cell cycle slows down a bit, and you can see it, then you will get it. So, long answer, I'm sorry, but it's a complex.
Les deux dernières questions seront celles de Vincent Collot et Claudine Junior.
C'est sur la paramutation. Sorry, on paramutation. So first, you know that paramutation is a plant phenomenon initially, and it happens in the F1. Do you have an effect in the F1 or not at all? At least
no, because we... do we have an effect in the F1? Well, no, actually, yes. Yes, we have, let's say, an effect which is transmission of either the active or inactive state, which is not fully penetrant, but yes.
So you can't see it in the eyes that well, but you know.
Yeah, you have it. You take okay. What you shouldn't do is lose homozygosity. But as long as the two are homozygous, in the F1 you do have paramutation.
And that's actually a related question. And so when you test for reversibility, then you make a cross between a naive and a paramutagenic or paramutable one. But you don't see paramutation here.
When you say it again?
When you test for reversibility of your state that you have reached at the end.
That's because you lose, as I told you, as soon as you lose homozygosity, you lose it.
Okay.
That's the reason for it.
Complicated.
So actually a single copy of this element doesn't do anything. It binds Polycomb, and it can silence transcription partially, but it will not be able to be strong enough to carry the information.
Last question, Claudine Junior.
You talked about the position of the transgene. Are these interactions requiring a position in cis or trans? And so does it mean that there are loops or just chromosomal interactions?
Okay, so in the case of the transgenes, they can happen in trans. In the case of these endogenous genes that we've seen it in cis, and we don't know that it can happen in trans. And actually, we have done IC very intensively to track for Polycomb-mediated contacts in trans, and they are quite sparse. So the case of the transgene, you have homologous sequence, and maybe the search is more efficient. But in the case of sequences that are different, they just bind Polycomb, but they have different contexts and other factors. I think it's less efficient, and you have more contacts with the cis flanking region. They can be far away; they can be several megabases away. But
thank you. I would like to thank all the speakers.